Hello everyone. Now I am going to take a seminar about the topic transmembrane transporter and also discussed about the ion transport, core transport, bulk transmembrane transport. The first of all we discussed about ion transporter. The transporter is a transmembrane protein that moves ions across a biological membrane to accomplish many different biological functions, including cellular communication, maintaining homeostatics, energy production, etc. There are different transporters included pumps, uniporters, antiporters, symporters. The active transporter or ion pumps are transporter that convert energy from various sources including adenosine triphosphate ATP, sunlight and other redox reactions to potential energy by pumping an ion up its concentration gradient. This potential energy could then be used by secondary transporters, including ion carriers and ion channels, to drive vital cellular processes such as ATP. The ion transporters are essential for proper cell function and thus they are highly regulated by the cell and studied by research using a variety of methods. Next, we discussed about a co-transporter or coupled transporter. The coupled transporter or co-transporter is a special type of active transporter, which accomplish import of different substances by a common or combined mechanism. The transmembrane transport of sugar, amino acid, etc. take place by um, coupled transporter. In some instance, the transport of these substances coupled with the active transport of sodium ions. The transmembrane transport of glucose is almost always coupled with the active transport of Na plus and it is carried out by common sodium co-transport mechanism. In this case, sodium-potassium exchange pump creates a steep concentration gradient across a plasma membrane with the active transport of Na plus out of the cells and the active transport of K plus into the cell. For the inward transport of every 2 K plus, 3 Na plus will be pumped out. Na plus probably activates the enzyme ATPase and thereby accelerate ATP hydrolysis to provide energy for the active transport of glucose. And next we discuss about the vesicular transport or bulk transport or cytosis. This is a bulk transport of large molecules and particles across the membrane. The material to be transported first gets into vesicles or pouch of the plasma membrane, which act as a transporting vehicle. The vesicular transport is of three kinds: exocytosis, endocytosis, receptor mediated endocytosis. The first of all we discuss about the exocytosis. Exocytosis is the explosion of cellular waste and export of some secretions products in large bulk from cell to the extracellular medium. It is common in secretory cells. The exocytosis is a relatively simple process occurring with the active participation of Golgi bodies. Next, we discussed about endocytosis. Endocytosis is a taking in the large amount of useful substance into the cell from cell from cell from extracellular medium. During its extracellular substance, the are first enclosed and then internalized by a portion of Plasma membrane. The portion of the membrane on contracting the substance undergo invaginations and forms pocket known as endocytic vesicle. The invaginations occur with the ATP utilization. Soon, transportation substance gets enclosed within the vesicle. The vesicle grows further inward, pinches off, and from plasma membrane gets completely internalized and forming endosome. It soon fuses with a primary lysosome and its contents are digested by lysosomal enzyme. The endocytosis is of three main kinds, namely phagocytosis, pinocytosis, micropinocytosis. First of all, we discussed about the phagocytosis. Phagocytosis is the bulk intake of large-sized solid substances. Phagocytosis of very minute colloidal particles is known as ultraphagocytosis. Phagocytosis is completed in three major steps, namely surface recognition and biting, membrane invagination, and vesicle formation. It is an energy dependent mechanism. It's all about phagocytosis. And next we discussed about pinocytosis. Pinocytosis or cell drinking is an intake of liquid and fluid substances, very minute particle. It occurs in eukaryotic cells which lack rigid cell wall. The inducer substance recognizes a specific receptor site on the membrane and binds with it. This is the followed by membrane invagination to form vesicle or channel. Finally, the vesicle or channel detach from the membrane and get internalized, drift into the cell interior and form pinosome. The pinosome will fuse with the primary lysosome to get its contents digested by lysosomal enzyme. The and micropinocytosis. Micropinocytosis is a transcellular transport of fluid substance bypassing intracellular components. It is highly specialized form of pinocytosis. It involves some steps. First, invagination of plasma membrane and formation of a micropinotic vesicle. Internalization of pinocytosic vesicle and formation of micropinosome. The movement of micropinosome to the opposite side of the cell through cytoplasm. The fourth one is the fusion of micropinosome with the plasma membrane as in exocytosis. Breakdown of the micropinosome, the discharge of its content. The micropinosome is a common endothelial lining of capillaries. The endocytosis, especially phagocytosis, is a specific or more or less selective process. It involves the contraction of actin and myosin filaments of the cytoplasm and ATP utilization. The third one is receptor mediated endocytosis. Receptor mediated endocytosis occur in specialized shallow depression of the plasma membrane known as coated pits. The cytoplasmic surface of these pits is lined by the coating material and formed mostly of a network of the membrane protein calithrin. The extracellular substance getting bound to the receptor called ligands. Ligands receptor complex get enclosed in the coated pits of plasma membrane. The pits now grow inward in large size, get heavily concentrated with the membrane bound ligand receptor complex and transform to coated vesicles. Eventually, they detach from the plasma membrane and get internalized. So, they lose their calithrin coat, merge together, forming a large calithrin-free vesicle called endosome. The ligand receptor complex eventually dissociate. The endosome develop a tubular portion and vesicular portion. The tubular portion contains ligand-free receptor in its membrane and vesicular portion contains receptor-free ligands in its lumen. The tubular portion now pinches off from the vesicle, gets incorporated with the plasma membrane to repeat the cycle. The vesicular portion fuses with a primary lysosome and get its content digested by lysosomal enzyme. The transport of cholesterol into animal cell is a typical example of a receptor mediated endocytosis. It's all about uh, transmembrane transport of transmembrane transporter, ion transporter, bulk transporter, and co-transporter. Thank you.